Welcome to the SAN Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Please like, subscribe, and share with your friends and relatives. It really helps support the SAN Science Experience. Today, we're embarking on a deep dive into a really surprising discovery about our immune cells, specifically macrophages. These are the sort of unsung heroes, constantly cleaning up debris, fighting invaders. We're looking at a groundbreaking paper titled Evidence that Mitochondria in Macrophages are Destroyed by Microautophagy. And it really redefines how these crucial cells manage their internal environment. Okay, let's unpack this.
Yeah, what's truly significant here, I think, is that this deep dive isn't just about some, minor detail in cell biology. It's really about uncovering a fundamental, maybe previously misunderstood way our immune system cleans house. This process, microautophagy, it directly influences how well our macrophages can respond to threats. So yeah, it's vital for our health.
Right. When we usually talk about a cell's internal cleanup crew, most people think of macroautophagy. That's the one with the double membranes wrapping around stuff. But this paper shifts our focus to microautophagy, which is more of a direct engulfment. And for a long time, scientists thought lysosomes, the cell's recycling centers, were generally, too small to just swallow up big things like damaged mitochondria.
Exactly. And that's where the big kind of aha moment comes in this research. They've discovered that macrophages actually have these specialized, much larger lysosome-related organelles, LROs, that are Rab32 positive. Think of it like finding a supersized internal Pac-Man inside these cells, almost 2,000 nanometers, quite large. And these LROs are just perfectly designed to directly engulf other organelles, including those broken down mitochondria. They even coined a new term for it, microautophagy. It completely flips our old assumptions about microautophagy in mammals right on their head.
Okay, wow. And here's where it gets really interesting for me. This new way of degrading mitochondria operates entirely independently, right? Independent of that common macroautophagy pathway, and even the ESCRT machinery, which is another sort of cellular toolkit.
That's right. It's not just tweaking an old system. It's like a completely different instruction manual being used by the cell. And the study pinpoints the key molecular players that make this happen. There's a protein conductor called Rab32 GTPase, and then there's this eat me signal on the damaged mitochondria ubiquitination that gets recognized by another protein, P62, which helps grab them for disposal. Oh, and PTN's 3A5P is involved too.
Okay, so Rab32, ubiquitination, P62,
yeah,
a whole specific cast of characters.
Exactly. And if we connect this to the bigger picture, this Rab32-mediated microautophagy plays a profound physiological role. It directly impacts how macrophages gear up for battle. That process we call M1 polarization.
Ah, M1 polarization. That's where they shift their metabolism, right? Towards glycolysis for quick energy bursts.
Precisely. They increase glycolysis and reduce reliance on mitochondrial respiration oxidative phosphorylation. And that requires getting rid of those damaged mitochondria efficiently.
Makes sense. And the evidence from the paper seems really compelling on this point. They made macrophages lacking this specific pathway, the Rab3238 double knockout ones, and those cells showed significantly reduced M1 polarization. Is that right?
That's exactly what they found. Their ability to produce key pro-inflammatory signals, things like IL-6, IL-1 beta, TNF-alpha, it was suppressed. And their metabolic shift toward glycolysis was impaired too. It really shows quite clearly that this unique microautophagy pathway is essential for proper immune function.
So it's not just some obscure cellular mechanism. It's directly tied to how these cells fight infection and inflammation.
Absolutely. Which naturally raises an important question for, future research and maybe even medicine. Given how crucial microautophagy is for activating macrophages, how might understanding this pathway, maybe even modulating it, open up new therapeutic avenues?
Ah, so you mean potentially boosting immune responses when needed, like in infections?
Or perhaps dampening overactive inflammation in things like autoimmune diseases by targeting this Pac-Man system, as you called it earlier. It's something to think about.
Definitely food for thought. So what does this all mean for you listening in? We've explored how macrophages use this distinct Rab32-mediated microautophagy pathway to get rid of damaged mitochondria. And this isn't just a tiny cellular detail. It's a fundamental process shaping their metabolism and their ability to protect us.
Yeah, it really deepens our understanding of the intricate ways cells degrade proteins and organelles. It reveals this precise, maybe underappreciated mechanism controlling macrophage function. It just highlights the amazing versatility of our cellular machinery. And like we said, it offers potential new avenues for research into inflammation, metabolism, maybe even broader cellular health. Could lead to new treatments down the line.
Fascinating stuff. If you found this deep dive as interesting as we did, please remember to like this video and subscribe for more insights into cutting-edge science. Thank you for joining us on the SANS Science Experience.